Kind of microbe.
Okay.
Barrier, presence of barrier.
And again, data interpretation.
I got all this in my notes.
I know you do. Next, be able to interpret data resulting from the experiment done in lab. Well, what was this experiment? 7-2, tell me what that is.
7-2
was that the caries?
Caries in the urine.
What happened to the disinfectant one
No,
that we
that's
did?
not caries in urine.
Disinfectant. That was on the last test.
7-2
Are we doing
was
disinfectants?
that's
That was the last test.
antibiotics. And
Oh,
disinfectants.
okay.
Yeah.
Is there any terminology to know there?
Disinfectant.
Do we
Oh,
do
yeah,
like
the
broad and narrow?
Broad spectrum, narrow spectrum. What about antibacterial, antifungal?
And yes.
What about what's it called? The disc or something?
Oh,
The zone of inhibition?
Yes, zone of inhibition.
Oh yeah, the circle around it. Okay.
Okay. And then the disinfectant experiment, that was a handout, not in the manual. This is
Like-
where you tested some chemical agent that is supposed to be antimicrobial. You remember you put them back. I put the bacteria on the pins for you, and then you
So
subjected the pins
It was like mouthwash.
for 5, 10, and 15-minute intervals. And so once again, we listed variables that influence how well disinfectants work. They included the exposure time, the concentration of the agent, etc.
Kinds of agent, concentration, exposure time, kinds of microbe. Those are the factors.
Okay? So now we're to 5.2 through 5.19, and these are those biochemical reactions. And so I can easily summarize what you have to know here. The handout that we used the second lab period of those 2 weeks, it's in chart form. There's 6 columns. The columns are the name of the medium. Now that's the inoculation period, Diane. There's the one. The name of the medium, what's important in that medium, what biochemical reaction are we looking for, end products, and positive and negative results.
Yeah.
That's it. And that was 2 weeks worth of lab. Now, if there's 2 weeks worth of lab, and Peterson makes the questions proportional to the amount of time spent, will there be more than 2 questions on these biochemical reactions?
Yes.
Yeah.
So will you
But
formulate
is
me
Peterson going to do that?
I do that intentionally.
Oh.
Because I swear on that lecture test there were when we had the invertebrates and the algae and the protozoans. That was like lab test 2.
Mm-hmm.
You asked one question on protozoans and like 17 on algae, and I was like, how is this proportionate? But I don't remember going over any of those in class, so maybe it was. Will you formulate me a hypothetical question on this? Here, I'll even
Sure. No, I know what it says. I wrote it.
Okay. I didn't know if you knew it by heart.
Yes.
Okay. Ready?
What biochemical reaction converts glucose into an acid and a gas?
Phenol red.
Listen to the question. What biochemical reaction, which column do you need to look in, Diane?
Oh, okay, the biochem reaction.
There you go.
The reaction
Oh,
part?
fermentation?
Fermentation.
Where do you see fermentation? I see HHHHR.
That's the second week,
Yeah,
Brewster.
you're on the
You're in the
wrong one
wrong
then.
week.
I
So
showed
you're saying
this to
look
you.
under the biochemical reaction, and that'll be
Sir, I
everything that it
just
does.
showed it to you, and you said, uh-huh.
Well, they all.
It is right for week 2.
Are you gonna want to know about the reagents too? The reagents?
Yeah, what,
Yeah, this one.
what reagent is used to detect indole production
Indole.
Indole, I-N-D-O-L-E. That's the,
Carbo blue.
indole is the end product from tryptophan hydrolysis. That would be week 2.
Are we still on the same page?
No. You're on the second, that's a question from the second week.
The one I was just on?
Yes?
Crap. I went to the other one.
I don't have any of that.
What reagent detects the presence of indole that results from tryptophan hydrolysis? Kovacs reagent. Amy's found it.
That's the sugar. Snyder almond.
Where are you?
Right here.
Ah, crap. I opened the same one again.
No, mine's printed out off. Right easy.
5252.
Sometimes hard copies work better than Brewster.
That's wasting trees.
This one?
This page.
Yeah, that's it.
No, that's inoculation
This one looks.
for saliva.
Yeah, you're
It should be 5-7.
So his question was.
One more time, 5-7 reactions. All right.
All I have on here is the orange. Oh my god.
I don't need to see this.
Got it?
Maybe. Looks about right.
What's the reagent that detects indole presence resulting from tryptophan hydrolysis?
You're asking for the reagent?
Yes.
That one? Kovacs
Yes.
reagent?
Yes. What are those scratches on your screen?
I threw it because I was watching a TV show and a bug crawled on me and I threw the bug and the iPad and it smashed up against the wall. And it costs a lot of money to get fixed.
I bet it does.
So I just deal with my cracks. They don't inhibit the usage. I might get a split
And the
trip.
bug didn't hurt you.
No,
Okay.
it wasn't the stinging kind of bug.
All right, next one. And this is the urine specimen. You need to know how to calculate the number of bacteria in a unit volume of urine.
You do it, you count the number of colonies times,
The number of milliliters.
the
Reciprocal
reciprocal.
of the dilution.
Mm-hmm.
Gives you-
Yep, it
And
go
that
like
get-
this.
Now that you have that number, next question. What's the significance of that number?
There's a range that if it's between those numbers,
Normal or increased risk or low risk.
Yeah.
Between 10,000.
If it's normal or what, Amy?
Or
If
you
you're
have
at
an
risk.
increased risk or a low risk.
for UTI,
Of a UTI. There you go. There you go.
teamwork.
All right.
Getting it.
We did something last
Now,
Formula?
that was just overall counts of bacteria. But then we used some special media and we identified certain categories of bacteria, like gram-positive and gram-negative and facultative and so forth.
For this exercise?
Yes, sir.
Further fermentation.
Yes, I got that. Hold on. We went over that. Yes, we did.
Lactobacillus
Remember, I kept saying
activity.
most.
Hey, can I tell the group something?
What?
And please don't take this in the wrong way. Some of you are doing now, maybe all of you, I don't know, should have been done a long time ago. You don't have your material organized. Organization is a key thing to getting this information in your cerebral cortex.
I agree.
But how would we organize it if we have, mine is in, chronological order from beginning of
That's
class
what
to
I
end of
do.
class. That's the only,
That's the list, that's the order there on this sheet right here, and you have this sheet.
but These sheets are all sorts of out of order. But maybe I should just hard copy them and then put them in order.
Yeah.
That's where I get lost, is because it's like
Hey,
5752 72
Rooster,
2871.
I'm not telling you how you have to do it. I'm telling you, you need to do it. And that doesn't just mean micro. That means any class you take. When you're 48 hours from the test, you ought to already have had it organized. Okay? All right. I'm not going to get any on your case anymore. Now, the next experiment was the saliva experiment. Okay? How did we calculate the number of lactobacillus in the saliva specimen?
Oh, the tube, the color, if it was green or if it was.
No. You're not listening to my question. How did we calculate the number, Pedro,
Cells per milliliter.
of lactobacillus cells in a unit volume of saliva?
Number of colonies times the reciprocal.
We did it
solution.
exactly the same way we did the urine specimen. But
Yeah.
now I'm asking about lactobacillus. How did we segregate lactobacillus from other bacteria that were in your spit?
We
Because
used a special.
the pH indicator.
Because the
was
medium was selective for lactobacillus.
Yeah.
Rugosa.
What made it selective?
It was acidic,
And how did we know it was acidic?
because the lactobacillus was able to grow and it's the only acidic bacteria in the mouth.
Mm,
No?
Yes.
Oh. And it was rugosa, selective.
There you go.
indeed, Snyder. Well,
So you got the answer. See,
one
Dana,
test tube, Snyder.
you see, if you look at that paper, you got all the answers there, don't you?
Yes.
That's why organizing it would help immensely.
Yeah, but see, I've just got Rugosa SL.
Selective.
Why don't
Okay, Diane,
I don't even know what the hell.
what does the SL stands for?
Selective,
For?
lactobacillus?
acid tolerant.
No, for lactobacillus.
Selective lactobacillus agar.
So what's special about lactobacillus?
It's acid tolerant.
It's acid tolerant.
Is there another word?
Now, are there other bacteria in the mouth besides lactobacillus?
Yes.
Well, what happened to them?
They can't tolerate acid, so they won't.
The acid killed them. So the only ones left were what, Diane?
Lactobacillus.
Lactobacillus. Okay. Then we calculated the population size, and what was the significance? What did the population number mean?
Bacteria, or is that
Rugosa is the auger, right? Rugosa is the auger.
Yeah, rugosa with the R.
What did the number mean?
The normal count?
Your high,
The normal
your chance
count.
of being susceptible to caries.
Cavity.
Okay. Now, what did we have to add to the data we accumulated that told us a rate of acid production?
How
Didn't we
fast?
add-
24 hours.
So time.
Time.
Time was the factor. Now this is on your handout sheet. The caries susceptibility
So,
experiment.
Less than 12 hours was your
24.
high.
24 hours.
It was first, wasn't it 12?
24. was the
24.
high, and 48 was medium, and 72 was light.
There you go.
More than 72 negative. Yeah.
Is that not on your
It
page
is.
there, Diane?
Mm-hmm.
Okay. All right, how to calculate the population significance, and the significance of the rate, okay? Now have a note at the bottom of this handout, this review sheet. For every exercise, if there was a special medium used, by special I mean it wasn't just plain old nutrient or tryptic soy agar, know why we used that medium, what was significant or special about it.
It was acidic.
What purpose did we use those media? Okay?
Mm-hmm.
And that takes us through that's the last one.
That's it.
What about the ones where we had the gas that produced acid? We don't need to know that?
That was the experiment that where
was the
Diane
bottom.
answered.
Fermentation.
Fermentation is what converts a carbohydrate into acid and gas.